Okay, so it looks like we have quite a few people here. I think I'm going to get started.
I cannot share my PowerPoint presentation with the share screen. I have to be, oh, now it's okay.
Yeah, you should be able to when it comes to your presentation. Okay. So let me start by saying good morning, good afternoon, good evening, depending on where you are, to our webinar i2Eye 2020, Innovative Imaging of Eye Disease. We're very excited to present the program from this year. And I want to thank the rest of the organizing committee listed here, who have been very helpful in putting together this exciting conference. So I wanted to kick off with a little historical note on the conference. It'll be very brief because we don't have that much history, but I think we've come a long way in a short time. We began in 2018 with a one-day course addressed principally to clinicians, centered around adaptive optics imaging, and we invited clinicians for practical training sessions on clinical AO devices at the hospital, and we included a small number of international invited speakers who are listed here. 2019, we expanded the course to a two-day multidisciplinary course, inviting this time imaging experts as well as clinicians, and expanding the topics to include OCT as well as adaptive optics. We again ran practical training sessions on clinical devices along with the scientific presentation program, and we had a larger range of invited speakers, international invited speakers who we were very pleased to welcome here in Paris. For this year's 2020 edition, we were hoping to be welcoming you here to the Quinze Vingts Hospital at the Bastille in Paris for a three-day course. Obviously, COVID had other ideas, so we've moved to the webinar format, which nevertheless has some advantages because we've been able to really increase the scope of the course and the number of attendees. So we're looking at a three-day multidisciplinary conference this time with a combination of clinicians, imaging experts, biologists, and computer scientists joining us here. We are not running practical training sessions on clinical devices this time, obviously, but we do intend to create a live online imaging training session and to put this onto our website around spring of next year. So we'll let you know, keep an eye on our website. We would like to still address the needs of clinicians to learn in practice on high resolution retinal imaging. So for the course that we're going to see over the next 3 days, we have a great program of invited long talks and submitted short talks. We actually have 50 speakers joining us here, which is really nice to have so many people from across the world joining us. We have 24 from France, 13 from elsewhere in Europe, 10 from the US, and 3 from worldwide locations. And many thanks to all of the invited speakers who are listed here, and of course to those who have submitted their work for presentation in the short talks. Thanks very much for everyone's participation. Just to give a brief overview of what will happen in future, next year we will be moving the hosting of the meeting to Ethan Rossi's group at the University of Pittsburgh, and it will be a collaboration between our lab and Ethan's lab. And if there is a real-life conference, not just a webinar, it will be in Pittsburgh. And then in 2022, we'll move back to Paris and so on. So the intention is to make this a bi-yearly occurrence between US and Europe. So just to give a very brief overview of the program, we're going to kick off today with some basic courses in biology, optics, and ophthalmology. This will be sort of courses for dummies style in response to some feedback we had from other years' courses. It's so that we can all get on the same page and make this a truly multidisciplinary meeting where everyone understands everyone else's area of expertise. We'll be following this up with the short talk sessions on photoreceptors and RPE. Day 2 will be devoted to vasculature, blood flow, and optic nerve, both in long and short talks. And then the final day will be the photoreceptors and RPE long talks session. Just to give a brief introduction to the Paris Eye Imaging Group. So this is a multidisciplinary group of clinicians, physicists, biologists, and computer scientists who've come together to develop new methods of retinal imaging in order to diagnose eye disease. And so we are developing prototype systems in the actual ophthalmology hospital of France here in the Quinze Vingts Hospital. And we're applying these prototype imaging systems to imaging in patients and then analyzing the results. So we really go all the way from histology through instrument development to clinical imaging in the hospital site. And we're located on the same site as the Research Institute, Vision Institute. And even though we cannot welcome you here to Paris, we wanted to show you and give you the opportunity to meet some of our team and view our imaging facilities. So I wanted to invite you to watch a short movie for the next few moments, and then I'll come back to kick off the conference. So let's get going on this movie.
The Paris Imaging Group is a multidisciplinary team bringing together ophthalmologists, physicists, computer scientists, and biologists in order to develop a new paradigm for clinical research and care. This unit was developed because we found that the diagnosis of retinal diseases was lacking behind the emergency in terms of public health. Indeed, there was a growing number of diseases despite the progress in therapy that requires new imaging approaches to better diagnose them and maybe better treat them. So we've been developing a collaboration with physics institutes such as the Institut Langevin and the Onera, and also the Vision Institute here, and the ISEP, an Institute for Computer Science. And the conjunction of these efforts led to the availability of new prototypes to explore retinal diseases at the cellular level, such as using adaptive optics, which we improved to obtain more than 100 images per second, OCT, which would improve to develop 3D imaging using the technology called full field OCT, and holography to develop the observation of the retinal blood flow. And the conjunction of these efforts with histologists who developed 3D imaging of human eyes, and also computer scientists to develop the extraction of new biomarkers, led us to understand better some disease, sometimes very common, such as age-related macular degeneration, but also more rare, such as retinitis pigmentosa, and even understand the complications of disease as common as high blood pressure, for instance. hope in the near future to be able to revolutionize the knowledge of retinal disease and hence help to develop new therapies.
So apologies if the video did not run correctly for some people. I had some feedback that it was not running well. So I will give a link out to be able to watch that video in your own time, should you wish. It's also available on our website. So just before we kick off the first session here, I need to give a few guidelines for the presentations. When you enter as a presenter for your session, please mute your microphone and turn off your camera during other people's presentations. When you wish to address a question to a presenter, please use the Q&A function. So this is a Zoom webinar which has a specific Q&A function for this purpose. And if you use this Q&A function, the questions will be addressed to will be sent to the session moderators, and they will pose the questions to the presenter on your behalf. So please do use that Q&A function rather than the chat function. All the presentations that we'll see will be recorded and stored for a month on our website. We'll be giving out a password to participants to access these recordings. And if you're a presenter and you do not wish your presentation to be included in that set online, then please do let me know by email. It's not too late. Presenters and moderators, when you attend the session in which you're presenting, please enter the webinar as a panelist using the specific Zoom link that you received to enter as a panelist. You can attend as an attendee for the other sessions. Okay, and so there concludes my introduction, and we're about to kick off the first session, which is this transdisciplinary course in biological, optical and medical aspects of retinal imaging. And the first presenter is Anna Verschueren from our group here in the Quinze Vingts, giving us some details about biology of the retina. So I'm going to stop sharing my screen and pass over to Anna just now. Is Anna there? Anna, are you able to share your screen to start your talk? Ah, there you are.
Yeah.
Hi.
Okay. I hope everything is all right. So yeah, okay. So hello everyone, I'm Anna Verschueren and I'm a postdoctoral fellow in the Groupe Hospital Paris and the Paris, inside the Paris eye imaging. And today I'm happy to share with you an introduction to anatomy and biology of the retina for non-biologists. My focus for today will be to give you the basic concept to navigate biology talk and papers on the retina. I will start by a quick introduction to the structure of the retina, then focus on the 3 different cell types you can find in that tissue. For every kind, I'll give you a bit of a biology 101 necessary for context and present a pathology where the cell type is especially affected. You'll recognize those by a change in the color of the logo and the title. I'm not sure. Can you see my pointer? Yeah, thank you. So without further ado, the anatomy of the retina. Most of you know already the anatomy of the eye is the following. the light enters through the cornea, passes through the iris and is focused by the lens and received by the retina. The retina is then responsible for its translation into electric signal understandable by the brain that the optic nerve will transmit to the central nervous system. My guess is for most of you, the retina either look like this, a bunch of layers with different intensity, either from optical coherence tomography or adaptive optics, or like this, a theoretical network of cells within a schematic. As a biologist with a specialty in histology, I have the privilege to see more details about the cells themselves and their delimitation, which is exactly the link we need to understand the retinal organization between these. The price is that I need to work on that before that. From a biology point of view, the retina is composed of 3 main cell types, which with highly different functions, organized both radially and tangentially to the retina. The neurons, the support cell, and the blood vasculature. If you quickly observe the structure of the retinal layer, we can distinguish 3 types of layer. The layer containing nuclei, the layer containing other cell components, and the membrane. The layer containing nuclei are the following: ganglion cell layers, inner nuclear layers, outer nuclear layers, and retinal pigmentary epithelium. In the ganglion cell layers, you will find ganglion cell, but also astrocyte and the superior vascular plexus. In the inner nuclear cell, you will find bipolar cell, amacrine cells, horizontal cell, and Muller cell. In the outer nuclear layer, you will find photoreceptors only. And in the RPE, retinal pigmentary epithelium, you will find RPE cells. On the other layers, the layers containing other cell components, we have 2 plexiform layers. Plexiform means composed of a plexus, thus a network of cells, cytoplasmic extension. We have the IPL and OPL, inner and outer plexiform layers, respectively composed of ganglion cell, bipolar cell, amacrine cell, and photoreceptor cells, bipolar cell, and horizontal cells. I know it's a lot. And those 2 layers both contain microglia and vascular plexus, respectively the intermediate and the deep one. The IS and OS layer cells are composed of the photoreceptors corresponding compartment, the inner segment and the outer segments. And finally, we have 3 membranes in the retina. They are basal membrane maintained by the cell and notably composed of collagen. They are the IML, internal limiting membrane, external limiting membrane, and Bruch membrane. Choroid is also really important for the retina, even if it's not, so to speak, a part of it, and it's juxtaposed at the end of the RPE. Now that the retinal composition is clear, we can go into higher detail. First, the retinal blood vessel, in which I will include the choroid. And before that, a bit of biology 101. Blood main function is to transport gas, nutrient, waste, cells, and hormone through the body. The arteries are the vessel that allow the heart to give the blood to the tissue. Capillaries are in charge of the exchange within the tissue, and vein will bring back the blood toward the heart. So arteries bring the nutrients, O2 and immune cells, and the vein will take care of the waste and CO2 and bring them back to the heart. These different types of blood cells thus present different structures in link with their function. At its most simple, in the capillaries, they are composed of an endothelium, which is a single layer of cell forming a tube, and a basal membrane around it. In more complex vessels, you can find multi-layers of smooth muscle, elastin fiber, and collagen. Their composition depends on the diameter and the function of the vessel. In the retina, we saw a moment before that we have the retinal layers and the retinal vasculature, and at the choroid, the retinal vasculature is composed of 2 to 4 vascular plexus or layer. They feed the inner retina that you can see here. They possess continuous capillaries, which mean that their capillaries limit the movement of cells and large molecules between the blood and the interstitial fluid, and they are responsible for the internal blood-retinal barrier. On the other hand, the choroid is composed of 2 to 3 vascular plexus, feed the outer retina, is composed of fenestrated capillaries, which mean that a large amount of molecule exchange can occur, and are responsible for the external blood-retinal barrier. Now, I twice talk about blood-retinal barrier because the exchange between the blood flow and the tissue are highly restricted in the retina. This tissue indeed has what we call a immunity privilege, which is a protection from part of the immunity mechanism of the whole body, meaning that the general mechanism of that immunity don't usually apply. The two blood barriers are composed by tight junction in between cells, which means cells are connected densely one with the other, and that component can pass in between the cells. Either the endothelial cells with the tight junction here in blue, or the RPE with the tight junction in the same color here. That means that all blood components reaching the tissue has done so by actively crossing at least one layer of cell, and in the most extreme case, 3 to 4 layers of cells. This ensures a high filter and protection of the retina. To focus on specific pathology where you can find damage on the blood vessel, we can focus on diabetic retinopathy. Diabetic retinopathy is a common complication in diabetic patients. We can observe microaneurysm, which are circular enlargements of retinal capillaries, microhemorrhagy in the pre-proliferative disease, which means in the earlier stages of the disease. In advanced stages, we can observe neovascularization, and sometimes, depending on the presentation of the disease, we can find a retinal edema, which are abnormal fluids accumulation within the retina, which lead to distortion in the retinal architecture. The current physiopathology hypothesis for explaining diabetic retinopathy are the following. Diabetes is characterized by an elevation of the glucose concentration in the blood. This is toxic for most cell types. Thus, it will have consequences to alter vascular permeability. The glucose and altered vascular permeability then promote leukocyte attraction and abnormal inflammation. The alteration on vascular permeability are because the high glucose will intoxicate pericyte, for example, and which will lead to the microaneurysm we talk about, the occluded and degenerated capillaries we talked about a bit earlier too. As the occluded and degenerated capillaries appears, the O2 concentration can locally drop, inducing a neoblood vessel proliferation in a pretty systemic mechanism, as in most of the body. When the O2 concentration drops too low, blood vessel tends to create new neovascularization. So now that we talk about blood vessel, we can go to the support cell in the retina. Support cells in the retina are composed of 4 different cell types: astrocyte, microglia, mural cell, and RPE. Their main functions are in the range of the glial cell function. Glial cells are the cell type that we can find both in the retina and in the brain, responsible for architecture, metabolism, homeostasis, and immunity of those tissues. For you to better understand, we can do a bit of biology about the inflammation. Outside the retina, when the detection of an abnormal component occurred, this will trigger a series of answers by the immune system. Recruitment of immune blood cell, both resident and from the blood circulation, activation of molecular cascade, for example, the complement, swelling of the area, and finally repair, often involving tissue scarring. Within the retina, the hematoretinal blood barrier that we talked about a bit earlier stops immune cells to migrate within the tissue normally. And there are other immunosuppressant strategies in place, which all in all avoid scarring and swelling in the retina, which would impair vision. So to go back to support cell. Support cells are divided between macroglia, microglia, and RPE. In the macroglia, you can find astrocyte and neural cell. Astrocytes are basically glial cells, but only for the ganglion neurons. They have a function of metabolic homeostasis and also a bit of human function. Neural cells are responsible for the architecture of the whole retina. Their presence is overwhelming, and almost all neural processes have a direct contact most neural processes only have a direct contact with each other. So like different neural cells will only contact each other at their extremity, and the rest is completely surrounded by neural cells, as you can see in the schematic I used here. They have metabolic recycling and homeostasis important function, and also human function. You can also have microglia, as we talked a bit earlier. They are the residing human cell of the retina. This is their main function. They can migrate and evolve to macrophage in case of local inflammation, which is a process called activation. And finally, the RPE. The RPE is a pigmentary epithelium, which means it is composed of a single layer cell and is sometimes named the pigmented retina. It's highly important in the proper function of photoreceptors, as it recycles components from the photosensitive cascade. And it also can have a role in inflammation. Pathology focus, the AMD, or age-related macular degeneration or disease. It's characterized with the appearance of drusen, which are detritus accumulation in between the both membrane and the RPE, and vision defect in the central vision. It can evolve into two advanced forms, the dry form or the wet form. Dry form is characterized by a degeneration of both RPE and photoreceptor in the central retina, and neovascularization show abnormal new vessels formed by the choroid. The current physiopathology hypothesis to explain AMD is that drusen came from all the accumulated physiological stress over the time, and that this physiological stress or oxidative stress is toxic and or overwhelming for the RPE cell. Because the RPE cell starts to malfunction, there are disruptions in the photoreceptors metabolism leading to their degeneration, which will then give the problem of vision, of course. And finally, the last cell type of the retina are the neurons, composed of the ganglion cell, the amacrine horizontal cell and bipolar cell, and the photoreceptors. But first, what is a neuron? A neuron, or a nerve cell, is an electrically excitable cell that communicates with other cells via specialized connection called synapses. They are composed of a cell body, dendrite, where they receive signal from other cells, often neurons, and axon, where it's transmitted the signal to other cells, often also neurons. The signal, as I call it, is an electrical change in the polarity of the cytoplasmic membrane. This electrical change occurs through the opening and closing of different types of ion channels in the cytoplasmic membrane. This is why in the neuronal network, you have an electrical transmission within the cell as a whole, and specifically in the axon, and you have then a chemical transmission in the synapse in between neurons. The neural network in the retina, I will only explain very briefly today, and in not that much detail, I'm sorry, but I don't really have the time to go deeper. The neural network can be artificially, for better understanding, separated in two different networks: the vertical pathway and the horizontal one. The vertical pathway is composed of ganglion cell, bipolar cell, and photoreceptors, and the horizontal pathway is composed of amacrine cell and horizontal cells. So photoreceptors are the cell transducing the light, meaning they transform a physical signal, light presence and intensity, in an electrophysiological signal. At this point, we can say that the signal is kind of analog. The higher the light intensity, the higher the signal. Bipolar cell transform the signal. Two populations exist, on bipolar cell and off bipolar cell. One activated by the light detection of photoreceptors and one inhibited by this detection. They lastly transmitted the signal still in an analog form to the ganglion cell. Ganglion cell translate the analog signal into what I would call a digital type, a thread of action potential, which is the common language of the brain, and send them through the optic nerve to the vision area of the nervous system, where it can be further processed. Thanks to the two bipolar cell population, the retina produced two different positive signals for both light and dark. So the signal is not ambiguous. That basic pathway is modulated by the presence of both horizontal and macrosynapse, as I said before. They allow other level of information integration which exist within the network. For example, the retina already processed color contrast and movement detection, and of course, intensity contrast. And I will also say that the shape and properties of the lateral pathway differ depending on the type of photoreceptors, rod versus cone, and the eccentricity, center versus periphery of the retina. And to focus on a pathology where photoreceptors are damaged, we can observe pigmentary retinopathies. Pigmentary retinopathies are a diverse group of pathology, often inherited or genetic. They present a loss of night vision, followed by a loss of peripheral vision, and pigment accumulation, which leads to abnormal pigmentation of the eye fundus. The current physiopathology hypothesis is a photoreceptor degeneration caused by a genetic defect on the photoreceptors of the rod first, led by the peripheral cones, probably because cone and rods are metabolism are linked one to the other, and the survival of one is necessary for the other. Photoreceptor degeneration then overwhelmed the RPE cells, leading for too much things to digest, which then degenerated, leading to a hematoretinal barrier disruption, which can then explain inflammation and blood vessel macrophage recruiting, which in the end led to more tissue damage. This is why we often observed macrophage in pigmentary retinopathies. In conclusion, I hope I helped you better understand both the anatomy and the physiology of the retina. Retina is a complex tissue with three main cell types: blood vessels, support cells, and neurons. I didn't talk today about it because we didn't really have the time, but the proportion and organization of those cell types vary tremendously along the eccentricity of the retina. And finally, all these cell types are interdependent. Most pathology will affect one cell type, which will have consequences on all of the cell types across the retina. Now I'm going to take a few minutes to present you my project within the ISTO Paris group. My main focus is the characterization of LC and pathological aging in the retina using immunohistochemistry, because we know that most ocular pathology are progressive and or degenerative. We need a better understanding of the LC evolution of the human retina anatomy to be able to understand the pathological evolution of the human retina anatomy. And we wanted to use histology as a reference for better in vivo understanding from adaptive optic and OCT. For this, we developed a human donor tissue bank, and we are currently investigating it using classical immunochemistry and new technology. Here you can see a preview of what we are working on. We have our objective is to be able to characterize all the cell type that you've seen today, the blood cell, the support cell, and the neurons. And we started by the photoreceptors. Here you can see an opsin staining on the cones, within a timeline. So 5 different donors of 5 really different ages. We already observed 2 really interesting phenomena in need of better characterization. A nuclear displacement, where cones nuclei migrate to the inner segment layer, and an opsin mislocalization in damaged cone. So I would like to thank you for your attention today. I want to give many thanks to all of my team and all the collaborator and platform we are working with, and you for your attention. And finally, I would like to encourage you both to keep in touch, here is my email, and to watch my teammate presentation on all the other great projects of the ISTO Paris group tomorrow at 5 p.m. And thank you again.
Thank you, Anna.
That was a very nice presentation. There was one question in the question answer to ask if there is a lymphatic system in the retina.
No, they're not. There are no. Thank you, Michel. Michel already answered for me. There are no lymphatic in the retina.
Yeah, it's been a hot topic recently, the lymphatic in the retina. If I may answer, I mean, maybe you wanted to elaborate, Anna, on
It's not at all my specialty, but if I remember well, the fact that there is no lymphatic in the retina is part of the human privilege of the retina, and I don't think I really know more about that.
I just wanted to add that it's a hot topic because it's been widely discussed in the brain, which is also controversial, in the mouse brain. And but I know from Florian Senlob, who extensively tried, maybe he's there, the panelist, not yet, extensively tried to, oh yes, you're there, Florian, so maybe you can answer about the lymphatics.
So yeah, people are wondering if there are lymphatics in disease, and there are publications that would suggest that after trauma, I think that there could be some lymphatics growing into the choroid, but that has been widely discussed, and it's not sure that apparently it's still the topic that we'll need a lot more data about. So there might be lymphatics evolving in disease or after trauma, but if you just look at a healthy eye, neither the choroid nor the retina has any lymphatics with the same markers they use to find lymphatics in the brain.
I think it had been widely discussed, especially in concerning subretinal new vessels, the participation of lymphatics, but it's not yet established.
Okay. Well, thank you, Anna. If there's no further questions on that biology topic, we'll shift on to the next presentation, which will be from Gilles Tessier, who is giving us a course in optical principles of microscopy for non-physicists. Gilles, you can share your screen. You ready?
I'm trying to, yes. Do you see it? Can you see my screen?
Yes.
Okay. So thank you. I'm Gilles Tessier. I'm in one of the teams of physicists at the Institut La Vision. So I'll be talking about microscopy for beginners, although I know that many of you are not quite beginners, to be honest. And here is a link if you're interested on to a beautiful website where you can find a lot of information on the basics of microscopy. And I have to say that I took many of my images from this website. So if I can switch, yeah. So we all know what a microscope looks like. So this is one of them. It's probably an Olympus one, I think. So it's a pretty complex system. Apparently, if you break it into its components, but actually inside it's quite simple. All you need to make a microscope are actually 2 lenses. So this is the basic sketch of how a microscope works. So the first lens here is the objective, which you will use to make a magnified image of this tiny object here, somewhere in there. So that's the intermediate image, which is magnified. And we all know that the human eye is more comfortable when it's looking at objects which are far away or at infinity. So you use a second lens, which is called the eyepiece, in order to project this image far away to infinity and have a look at it comfortably with your eyes. So two lenses are enough to make a microscope, but actually nowadays, most, if not all, microscopes actually use three lenses. So the first one has been divided in two. The objective now forms images of the object, which are at infinity here. And you use a second lens called the tube lens, which is typically 16 or 18 centimeters in focal length. And this tube lens forms an image which then, in much the same way as what I said before, will be looked at using an eyepiece. And the advantage of using this system here is that if you look in this region here between the objective and the tube lens, which is inside your microscope, you have a region where all the beams are actually parallel and collimated. And this is quite important in modern microscopes because you always want to insert new systems into your microscopes, for instance, dichroic mirrors or filters, which are very sensitive to the orientation of the optical beams. So having this parallel or infinity space here to work with is quite convenient when you want to insert accessories in your microscope. So this works beautifully, as you know, to magnify images, but having magnification in microscopy is clearly not enough. In most cases, what really matters is actually resolution. Of course, contrast and selectivity are very important too, but resolution is probably the essential parameter. And in most cases, if you have a good microscope, diffraction is actually the limit to the resolution that you can obtain. So let's have a look at diffraction, and don't worry, I'll just be using a very basic Huygens Fresnel principle here, which says that if you have a wave such as the one that I'm showing here, you can draw wavefronts here, which are these planes here in the case of plane wave. And what Huygens Fresnel principle says is that every point on this wavefront can be considered as a secondary wave source of small wavelets. So you've got tiny sources everywhere across this plane here, which will propagate, and this is how you get your next wavefront, and so on and so on. So this is fine if you've got a plane wavefront, but what happens if you have, say, this point source, this light bulb, and somewhere in your system, your microscope, you have an aperture here? Well, what happens then is that these wavelets here will also propagate to give you a new wavefront. But if you look at what the light path should be with these red optical beams here, you realize that on the edges you have light in regions where you shouldn't have any light. And this is diffraction. So if you look here on the right at the shadow basically of this razor blade, well, you see on the edges diffraction and interferences in this case gives you this this light and these dark regions, or you can't see probably the light which should be in the shadow here, but there's light where there shouldn't be any. And actually this is the main problem in microscopy. So I'll try to show you an animation. I'm not sure if you'll see the screen because it's in another panel here. Can you see the other? I hope this works. So the idea is to show you what happens when you change the wavelength of your illumination here. So if I go to the blue region of the spectrum here, you see that the amount of the diffraction is reduced. And if I go towards the red, you've got more diffraction here as the wavelength increases. And in much the same way, if you decrease the size of this aperture, such as here, and especially if this aperture has a size which is of the order of the wavelength, there is light almost everywhere. Light is diffracted in many directions. And this is crucial in microscopy because there is actually an aperture in every microscope. So I'm coming back to my slides here. Yep. Yeah, so here's what happens when you have a slit such as this one. So light is diffracted, especially if the slit is small, if the aperture is small. And actually in a microscope, you always have an aperture because you are using lenses which have a finite size, such as this one. So for essentially the same reason, if you use a small lens, you use a small aperture and you will have lots of diffraction. And conversely, if you use a large lens, such as this one, it's equivalent to having a larger aperture and the effect of diffraction will be reduced. So this is essential. You want to be in this second configuration here to have to be able to focus light properly. Actually, what matters is not so much the size of the lens or the aperture, but the angle here of the light that you can send towards your sample or collect from your sample, which is typically called U or alpha. So this is the main parameter here, which will define the amount of diffraction that you will have in your microscope. So these are the consequences of this diffraction in imaging, which are related to what is called the numerical aperture here. So here in this sketch, you see the angle U and the refractive index N of the medium in which you're working. So people define this numerical aperture parameter N times the sine of the angle U here. And as you see here, the larger the angle, the smaller the spot on which you'll be able to focus. And conversely, if you have a small angle here, you will not be able to focus light properly. If you now look at the effects of this in imaging, quite clearly here you have a poor image of these small beads here, which is poorly resolved. And if you increase the angle U, if you increase the numerical aperture, you have a better image in which you clearly resolve these particles. And this limits the resolution of your microscope. As you see here, well, the size of these airy discs clearly has a consequence on the resolution, because if you try to look at two objects which are close together, such as the ones here at the bottom, you realize that if you get these airy discs too close together, this gets blurred and you can't see you can distinguish these points anymore. The limit is actually exactly the size of this airy disk, of this diffraction pattern that you see, which can be calculated. It's 1.22 lambda over NA. So lambda is the wavelength, NA is the numerical aperture of your microscope objective. And this 1.22 factor is actually what you end up with if you do the math correctly, but it's not really interesting, and people typically use this factor. This is if you use coherent light, such as a laser, but if you use incoherent light, there's actually a factor of 2 better, which means that you will always have a better resolution if you use non-coherent light. So basically, this is carved in stone, quite literally. This is a picture of the tombstone of Ernst Abbe, who demonstrated this diffraction limit in microscopy. And this limit has been reached quite early in history. Around 1886, people were already able to make microscope objectives which reached, just as well as we do today, the limits of diffraction. So essentially, microscopy didn't move much for more than a century. What could you do? You could, of course, improve the design. So having better corrections to have better resolution and get as close as possible to the ultimate limit to the resolution. You could also reduce the wavelength, because that's this lambda term here. So going to the blue or the UV, if your sample or your study is low, at least. And the last thing you can do is to use immersion. that is to increase the refractive index here. So clearly this is on the below the fraction bar. So if you increase the index, you will have a better resolution. And this is all about immersion objectives. So the idea is that between your objective and the sample, you can add a drop of water or oil, which will increase the refractive index in which you will work. And as shown on this diagram here, you probably know that if you're underwater and look towards the surface, the surface of the water will behave like a mirror. This is because most of the light will be reflected off water if it's at a large angle here. So if you're working in glass, the same thing happens. Lots of beams will remain trapped inside your sample and will never escape the glass slide to go to your microscope. So here I have a second animation to show you this, just to illustrate the fact that if you use a higher refractive index in your medium, you will be able to collect more light. So this is what happens in air here when the refractive index is 1, and these beams stay in the cover slip of your sample. But if you change the medium, if you add water around an index of 1.3 or something, then you see that these beams now are escaping the glass and can be collected. And if you increase it even more to 1.5, which is the refractive index of oil typically, then some of these beams can even be collected by a very classical lens. So you're collecting more light, of course, but more importantly, you have increased your resolution. So this is the idea behind immersion. So again, around the beginning of the 20th century, microscopy was essentially considered a solved problem. I've got this old paper, which was read in 1886, and in which people say that the limit of microscopic vision has been reached and no further important improvement is possible. And they were absolutely right. Actually, going beyond the diffraction limit amounts to breaking the uncertainty principle of Heisenberg. This is not really something you want to mess with in physics. Well, actually, it's possible, and things have evolved quite a lot in the recent years. And some people have shown that a super resolution beyond this diffraction limit is possible. I'll be talking briefly about palm or storm microscopy and STED microscopy. And then to list some of the main improvements, well, in my opinion, at least in microscopy, I'll talk a bit by 3D microscopies, and finally about quantitative phase imaging, which is a nice way to go beyond this. So this is all about in vitro microscopy, of course, and I think I refer you to a bit later to Serge Meimon's talk about adaptive optics, in which I guess he will show you ways to beat optical scattering, diffusion, and aberrations to improve the resolution in living tissues and in vivo. So about super resolution first, this is typically an image which is just filaments, microtubules, which are stained in a classical microscope. And this is the kind of image that was obtained by super localization by these two guys. The three of them got the Nobel Prize in physics in 2014, I believe. So Merne and Betsy here were able to use super localization to obtain this image, and I'll show you later some STED which was proposed by Stephen Heller. So the idea of super localization is quite simple, actually. If you assume that you have a single molecule which is able to emit light here, as we said before, the best image that you can get from this molecule is this airy diffraction pattern here. So you're lost because you have a tiny object, but you have a very large response or very large image of this tiny object. But there is one thing, which is that you can actually calculate the center of this pattern with a very good precision. You can calculate the center of mass of all these pixels and say that I know that my molecule is exactly at the center. It's here. And this can be done with an excellent precision. It's actually in the nanometer range, or it can be pushed, of course, but you can localize your object with a beautiful precision. So this is the only idea you need then to make microscopy. Suppose that you want to image this structure here with a classical microscope. If it's too small, the image that you would get would look somehow like this. And these are slides by my colleague Marcel Lauterbach. But now the trick is, what if you can switch on one by one the molecules which are inside this pattern? Switch them on between a bright and dark state. This is possible using photophysics. You can turn a molecule on and off, make it fluorescent or dark. And then if you switch them on one by one, typically this is done using a molecule which is called the activator, which allows fluorescence to happen. And then you have a second laser which pumps the fluorescence of the molecule. And you can really drive fluorescence on and off using this kind of cycle and these molecules. So you have one molecule here which is on. You have this airy pattern on your camera, which you localize here, and then you store in memory the position of this molecule. And you do this again. Here, you're not lucky. You have two molecules which are close together. You have a broad response. You're not able to distinguish them, but well, never mind. You just store the position and do this again and again until, well, you guessed it, you have a pointillist image of the thing you're trying to image. So point by point, you're able to draw an image of the structure that you are trying to see. And again, the resolution that you obtain in this case is essentially driven by the accuracy with which you're able to localize your molecules. So it's not infinite. There are limits to this accuracy. Essentially, it's about photon noise here. So the more photons you have, the better your accuracy will be. But there are other factors. The pixel size of the detector, the noise of your camera is also important. The background of your image, the number of scattered photons, and also, of course, the numerical aperture. If you start with a nice image of the molecule, it's always better than a bad one. But essentially, the main parameter is signal-to-noise ratio. And of course, it's not always very good. If you're looking at a single molecule or a small object, you don't have many photons quite clearly. But still, you can have, if you optimize this signal-to-noise ratio, you can have a very nice nanometer range precision in this pointillist image of your objects. I'll now move to another super resolution technique, which was introduced a few tens of years ago, STED, in which, well, it's actually, deterministic microscopy. I mean by that in the Palmar storm technique, which I just discussed, you have no choice but to hope that you will have points on your structure. Here, using STED, the nice thing is that you can choose where you're looking at. So it uses fluorescence, basically. So you have to tag your sample. But the idea is that instead of letting it emit as much fluorescence as it wants, you will use stimulated emission, which is basically the process used in laser, to force the molecule to emit its light. And once it's been forced to emit light, it cannot fluoresce anymore. So the idea here is that you excite a region which is, of course, diffraction limited. So it's a classical poor resolution, but then you use a ring-shaped beam here to de-excite the molecules on the edges. And only the molecules which are exactly at the center here will be allowed to emit fluorescence. And this is really the trick. If you reduce this region, you will only interrogate a small region of the sample, and then all you have to do is scan your sample to make an image. So this is what the system looks like in principle. You use this one this blue beam here to excite the fluorescence, and this is the image that you would obtain. And then you've got the second stead beam, which using a phase mask, which looks like this. This stead beam, which is a donut-shaped region. And as you see on this animation, if you increase the size of the donut, you will only have a tiny region which is not illuminated by this stead beam. You will only have a tiny region at the center, which will still be able to emit the fluorescence that you're looking at. So again, the precision, the resolution that you can obtain here, which is driven by the size of this small region, is not infinite. It's again essentially a matter of signal to noise, but also the amount of light that the sample can withstand before it's eventually bleached and destroyed. But still, the images that you can obtain with this are beautiful. This is a comparison of Vimentin filaments with a confocal, so already a good microscope. And this is a stead image of this same region here. I also have images by my colleague Marc Guillon here, in which you see living neurons, and these are synaptic buds which are ready to project other dendrites here from the neuron. And again, the main advantage of this microscopy is that you can really choose where you scan and where you make your image here, and you do not depend on the random position of fluorophores. So I'd like to move to a few 3D microscopy techniques. Actually, just one of them, which is probably the most important, well, one of the most important today, which is light sheet microscopy here. And the idea is to illuminate just a plane of your sample in order to be able to make some cross sections in your sample. And by moving this thin sheet of layer of light, you'll be able to excite fluorescence in different planes and image it perpendicularly using another objective lens here on the side. So as you scan this green beam across, you'll get an image. So there are several ways to do this. One is, as I just discussed, to create this thin layer of light, which is done using a simple cylindrical lens. So really the kind of lens that you would use to treat astigmatism. But there's another one, which is to use what are called Bessel beams, which are beams which remain focused over a very long length, at least longer than what you can do using a classical illumination, such as shown here. If you actually shine a ring of light onto your microscope objective, the region in which it stays focused is longer. So basically, in both cases, the idea is the same. You just interrogate a 1D or a 2D region of space and then scan it in order to make a 3D image. To create these Bessel beams shortly, it's quite simple optically. All you need is a cone-shaped lens here, which will help you focus your light into this very narrow and long region here. So I'll just show you some classical images here. This one is an animation of an embryo here, which was obtained using a light sheet by these guys here. So very nice and impressive images, at least for a physicist like me. And here, well, just to say that this kind of technique is very adapted to cases in which you have no or little optical scattering. So it's a good combination with the clarification process, in which well, you erase basically the refractive index contrast in your sample to make it transparent, which allows you to shine these light sheets across the sample. Again, images of a clarified brain here. And these are the kind of 3D images. It's a pity I can't move them here, but really by scanning plane by plane, you have a 3D stack of images in this mouse brain here. And the last image, which you briefly saw in the video, I think, which was done at the Paris imaging group, of a clarified mouse eye, which was obtained using this kind of clarification and light sheet imaging. So finally, I'd like to say a few words about quantitative phase imaging, or phase imaging to start with at least, because there's a lot of information in the optical phase. The idea is that if you send a plane wave on a cell here, the waveform that you will obtain will actually be distorted because light has traveled through more or less matter on its way. So it will be delayed or advanced depending on the amount of matter it's crossed. But if you look at this in intensity, there's not much to see. Having a phase sensitivity is clearly very interesting in this case if you want to image this kind of objects. So this is well known, at least in the qualitative version of differential interference contrast, which was proposed by Fritz Zernike, for which he got a Nobel Prize. You just use a small tool, which is basically a faceplate to have a twin image, which will interfere with the image that you would normally have to create this kind of patterns here in black and white and give you a label-free contrast. But it also works in colors, and you get these kind of beautiful images here using this contrast. But it's not quite phase. Differential interference contrast is not really phase contrast, and it's not quantitative. You cannot say how much dephasing you had in your sample. And this is important because if you're able to, as shown here on this, on the image at the bottom right here, if you're able to measure, to image the phase, there's a lot of information in there. And essentially, there's information about the amount of dry matter in your sample. And in this image by Pierre Bon from Bordeaux, he was able to measure quantitatively the refractive index change, which reveals the amount of dry matter, which means everything except water in this sample. So really, you can make quantitative measurements using this, and more interesting, you can also reconstruct 3D information and have a 3D reconstruction in your images. So I'll just show you. Sorry. A way to image the phase, which we developed in our team very quickly, which is quite simple since it uses a simple diffuser such as this thing here. So basically, it looks a bit like a piece of Scotch tape. The idea is that if you try to image through this diffuser, the pattern that you will have will look something like this. So it's a random, nearly speckle pattern. And if you change something, it should change the phase in your sample. I hope you see this blinking here. What happens is that you distort the phase, and this will move the pattern slightly. And my colleague Pascal Beto devised a way to track these movements and to turn these into phase images here, such as the ones you see here. So this is a live image in which you see essentially the mitochondria moving, and this is completely label-free imaging. And I'd like to wrap it up by showing you what I think is a very nice application of this phase imaging by Pierre Bon in this paper here, in which he shows that phase imaging in thick samples, this thing is 100 or 150 microns across, he's able to super localize fluorescent images and to make super resolved imaging out of this. He demonstrated a resolution of 50 nanometers deep inside tissue. And this is because phase imaging allows him to localize these fluorescent objects with an extremely good precision. So I think it's really a good summary of all the different aspects and developments I've shown you in microscopy. It's 3D, it's super localization, super resolution, and some phase imaging. And I'll finish with this. Thank you very much for your attention.
Thank you very much, Gilles, for this really nice and comprehensive talk that taught us all a lot, I think, about microscopy. Did anyone have questions for Gilles? We have a couple of moments.
No. Okay, so maybe I'll go on it with my talk if there's no question.
Sure. So the next presentation is from Michel Paques on retinal diseases for non-ophthalmologists. Thank you again, Gilles.
So, hi everyone, I hope the sound is okay. Apparently it's okay, if nobody says it's bad. Okay. So I'll talk a little bit about clinics today. It's difficult to teach ophthalmology in half an hour. I decided to focus on a specific disease, which is age-related macular degeneration, and how does the progress in retinal imaging helped us to understand the disease, and including recent research developments. So, So first of all, how can we do a fundus image? In fact, most of you, probably all of you, have already seen a fundus even without noticing that it was a fundus image. When you do a fundus photograph, especially with a flash in a dark environment, it's very frequent to have the red eye phenomenon, as you see here on the right image. It's not visible on the left image because the pupils are not dilated. And the red eye, in fact, is the reflectance of light from the fundus, from the black of the eye is transformed into red. That's because the eye is a good mirror. Well, not a very good mirror, but a mirror sufficient enough to send back some of the light it sent. And you see it because the light you send into the fundus, which is from the flash, has a very narrow angle with the light received back, and that you see with your eyes. So when light is coaxial to illumination and the axis of observation are coaxial, then you generate the red eye phenomenon, and this is more accentuated when the pupils are dilated. That's why usually there is a pre-flash, send it to close your pupil, and then take the image, which minimizes the red eye phenomenon. Well, nowadays it's mostly done digitally, but if you want to do it, let's say, the good old ways, it's quite simple. You just have to increase the angle between observation and illumination. So the eye is interesting in many ways, not only for optical reason, but also, of course, for medical reason, because it's an organ, of course, of important. Vision is probably the most important sense, but also because it's a window on the brain. That is, many structures seen elsewhere in the body, and especially in the brain, are encapsulated in a relatively small volume that is optically accessible for many of these tissues. So this is figuring the pathway of light through the eye. So the basic anatomy of the eye here, the cornea, the iris, and the lens here, which are the main purpose of these systems is to focus light accurately to the retina, this layer here, and during this talk, we'll focus our interest on this small area here called the macula. This is a magnification on macula, and the light has to go through the different layers of the retina that you see here to be captured here by the photoreceptors. So retinal imaging have been the basis of diagnostic in ophthalmology for now nearly 200 years. And this is because the light going on the eye is either captured by photoreceptor, either dispersed on the fundus, but a little proportion of it comes back and is reflected back in, let's say, 1 photon out of 10,000. And this small proportion is enough to have a lot of information about the structure of the retina, and the quality of that information increased over years. So this is the image of a normal fundus. Here on the right here the optic nerve. Here you have the vessels, the artery here, a little bit more clear than the vein, the darker veins here. This difference in coloration is due to the fact that the oxygen content of arteries is higher. So saturated hemoglobin, is lighter than the desaturated hemoglobin. And the color of the retina is mostly orange, pink orange or yellow orange, because of the presence of a pigment in the retinal pigment epithelium and the choroid. But it's not, what you see is not the retina, in fact. The retina is mostly transparent using this kind of approach. You see here some reflectance at the surface of the retina, the correlation of the background layers, the choroid, but not the retina itself. Most of their interests here in ophthalmology in general, but in this talk in particular, will be centered on the macula, the central part of the retina, which bears the highest visual acuity, because the concentration of neurons is the highest in that area. And whatever happens in the macula may directly affect vision and directly affect quality of life. You may have large lesions in the periphery of the retina that are not really damaging vision. And even sometimes large lesions in the periphery of the retina are not noticed by a patient. It's when the closer it gets to the macula, and especially to the center of the macula of the fovea, the more annoying it may be for vision. Here you have a case of a diabetic patient. You can see it's let's say I know it's a diabetic patient, of course, but it's typically presenting under the form of these red dots here, which are blood, these yellow-white dots here, which are lipids accumulating onto the macula. So we can say that this is probably the fundus of a diabetic subject, showing some lesions of vessels, creating these lipid accumulation into the macula and probably affecting vision. So from fundus color photography, we can do a lot of diagnosis, very precise diagnosis, even if we cannot quantify exactly what we observe. And we don't see the retina itself. Other features that can be seen are, for instance, hemorrhages that you see here in the case of retinal vein occlusion. And it's probably due to the fact that there is a circulation abnormality here, something somewhere in the optic nerve, because through the optic nerve, the evacuation of the venous flow. So when it's blocked for some reason, we don't know, in fact, the cause of these central retinal vein occlusion. There is an upstream dilation, the blood still goes in the fundus, but cannot go out. But most of the talk, as I said, will be concentrated on dry age-related macular degeneration. You probably heard of that disease, which is the most common cause of visual loss in industrialized countries, at least. More common in Europe and in the Western world and in Asia. So this is quite common. It may lead to very severe visual loss. There is no treatment. You know that there's a strong genetic component of the disease, but despite many discoveries in the cause of the disease in the last 10 or 20 years, we still cannot stop this specific form of age-related macular degeneration. I will not talk about the second type of age-related macular degeneration, which is the wet form of age-related macular degeneration, which is presenting another way, but still affecting the macula, still the same people, still the same genetic component. There is a treatment for that at least can stop the disease. I'm talking about the other form, roughly the same frequency, that's characterized by extensive lesion during years and years, and there is a loss of vision occurring in the center that expands progressively over years. Physically, these lesions are quite small, a few millimeters at best, but so why such a small lesion damaging so much vision? Well, it's because, as I said, in the center of the vision, every photoreceptor counts, because the area of the retina with which you are reading the letters on this slide, the area measures about 300 micron. It's the fovea. And this is the area with the best visual acuity. And when you go far from the fovea, the visual acuity decreases regularly because there's less neurons. There are also some image processing that concentrates the signal from several neurons, which also contributes to the loss of visual acuity when you go in the periphery. So whatever happens in the fovea is exquisitely important, and any small progression of lesion in the fovea may lead to dramatic loss of vision, while if it plays in the periphery, it may not affect vision at all. So age-related macular degeneration, the dry form, is progressing over years, and we have a quite good knowledge of the natural history of that disease. We can predict to some extent the presence of age-related macular degeneration in some patients, because it starts in aging patients, let's say over 55, by the appearance of these yellow-white spots here that are called drusens. They are a sign of abnormal aging of the retina. It's abnormal aging, even if it's quite frequent in the population, several percent of the population. It's not necessarily leading to age-related macular degeneration, but it's a risk factor. And this is not the same patient, but it illustrates you the course of the disease. It happened in both eyes, and that's the dramatic problem also. It's not necessarily the same time in both eyes, but it may affect rapidly the second eye when the first one is affected. And this is, at this stage, vision is usually completely normal. Vision problem starts when you have this spot here, which means that there is a loss of cells in the center, especially pigmented cells. That's what we call say it's a disease causing atrophy of the retinal pigment epithelium, the background layers of the photoreceptors, I said, and I in the first talk. And this spot, this atrophic spot, will expand very slowly over years and may eventually reach the whole posterior pole, and you can imagine that vision is already severely affected here, but here it's even worse. You're not completely blind when you have dry age-related macular degeneration, but you may be very severely affected and sometimes not even able to walk securely in a place you don't know. And it's a major cause of distress for the patient because they see every day, sometimes every day, the scotoma grow, and it's quite distressing. So what did we know about age-related macular degeneration before the advent of modern imaging? Well, we had already histology, and by histology we know that the disease is affecting the outer retinal layer, not the inner retinal layer, but the outer retinal layer, the photoreceptors, and retinal pigment epithelium. On fundus photograph, you don't see that your photoreceptors are missing, but you can see that RPE is missing. This is why they give this fundus appearance. And this is a scheme summarizing what we observed with the frontier between the normal-looking retina here, the margins here, and the area of atrophy here, corresponding to the successive area here in the center, the margin, and the normal periphery. So the patient can still see with that area. So one of the problems of the disease is understanding how does it grow over time. How does it appear that we know it, inflammation is involved? How did it grow is still a matter of speculation. So let's start with drusens. These drusens appear under this form of little whitish, yellowish spots dispersed in the fundus, often concentrated in the macula. And before modern imaging, we have very little information about where they were actually. And at the advent of imaging, and especially the combination of multiple approaches, what we call multimodal imaging, that help to understand what's happening in these patients. So this is an illustration of multimodal imaging. We can combine illumination by infrared light, by blue light, we can do autofluorescence in blue and infrared light as well, color fundus photograph, of course, and we'll talk about adaptive optics and OCT as well. So currently, doctors combine these information to understand what's going on in that disease. And this is here the close-up of a patient with age-related macular degeneration here, and you see here the atrophy, which appears by this modality, it's infrared light. Each modality gives a different information. In infrared, it appears as a white lesion. Normal retina is here, And this is a close-up here, and the arrow shows a pigmented depot here that also appears white. That's why we cannot distinguish solely based on one modality of imaging what lesion it is, what type of feature it is. We need to combine information from different sources, and if you do infrared autofluorescence imaging, then you more clearly see the difference between the atrophic area and the spot here. And since it's fluorescent in infrared, it's probably due to melanin, which is the main pigment showing infrared autofluorescence in the fundus. Here we see the color fundus photograph, and what we have seen here and here is brownish. You see it here also and also, which confirmed the hypothesis that it's pigment deposition. We'll talk later on about higher resolution imaging, such as adaptive optics, and it's OCT that will also be the subject of this future. This is OCT. So then came OCT, and OCT, which is a technique based on interferometry, has revolutionized the way we look at the retina, because with OCT, finally, we had access to the retina itself, to normal retina. We can see the abnormal retina when there's deposit or accumulation of something, but the normal retina was inaccessible or mostly inaccessible until OCT came, and currently we can have a very nice view of the different layers of the retina. And here in this slide, you can also have the structures of the choroid. The different layers here correspond to different substructures of photoreceptor. Maybe the eye is probably the only organs in which you can distinguish subcellular structures, and especially in this case of neurons, so the really important cells. This is a superimposition of OCT and of histology, showing the very nice correspondence between the different layers of the retina and the OCT. So let's go back to the drusen we saw. So what is it showing by OCT? So by OCT, we clearly show that these are located in the outer retina again, and we can see the volume of these drusens and with the accumulation in the macula, and you can see that the overlaying layers are more or less normal. The outer nuclear layer maybe is slightly thin, but overall the structure of the retina is rather preserved. The drusen, now we know since recent work by combining OCT and histology, that drusen represent a complex area of phenotype with probably different mechanical, different origins. Some drusens may be over the retinal pigment epithelium, some may be under the retinal pigment epithelium. They have different denomination. It's still it's an area of research still actively going on. And all these drusen or drusen deposits, whatever the origin, are linked to aging, and they are predictors of the occurrence of age-related macular degeneration, even if the link between drusen and age-related macular degeneration is not very clear. So to explore age-related macular degeneration, you need to use multimodal imaging as shown here. So as a reminder, I put here the normal macula. And here on the line beyond, we have the different aspect of the same eye with different imaging modality, color fundus photograph here, blue autofluorescence here, infrared imaging here, and OCT. And by color fundus photograph, in fact, it's probably the less informative, one of the less informative modalities. In fact, we have much more information. Well, maybe it's because I need better use of it. By blue autofluorescence, for instance, because blue autofluorescence reveals pigment contained in retinal pigment epithelium, which is called lipofuscin. So when it's black, it's because there is no lipofuscin. It's supposed to be grayish because lipofuscin is more or less diffused in the RPE. So when you lose the RPE, you lose also the lipofuscin, and so the black spot appears. On the opposite, you may have hyperfluorescence. So this makes you detect some accumulation of fluorophores, maybe lipofuscin, which are not detectable by on the color fundus photograph. Lesions are very easily seen, also very well seen by infrared reflectance imaging, which have the advantage of a color fundus photograph that it's not sending too much light to the patient. It's much more comfortable to the patient to have infrared imaging. So that's also important to in clinical practice, you have to use imaging modalities that are better tolerated by the patient. Infrared imaging, for instance, can be done in non-dilated pupil. You don't need to dilate the patient to have a good quality image. While by color fundus photograph, usually they have shorter wavelength, and it's commonly necessary to dilate the patient, especially if he has, for instance, cataract. So, and finally, you have the same lesion seen by OCT here, which is infrared light again, so very well tolerated. And you have a very, you can compare this, the aspect of this central atrophic area here by color fundus photograph, blue autofluorescence, infrared, and OCT. Currently, the OCT is becoming the reference because it gives out not only the size of the lesion, but also structural information on the overlying retina, which is not visible by the other modalities. An interesting feature is to see the OCT, for instance. You see this is white in the cord beyond the atrophic area, and it stays darker here. This is because the pigment here is still present and not here. So incident light can go through and reach the cord and come back and give a better signal from the cord than the area still covered by the retinal pigment epithelium. So the subtle changes in the histological structures of the retina can be readily well detected by imaging. And that is also a unique opportunity in ophthalmology. We can have very fine histology information just by analyzing the light coming back from the fundus in different ways. In more complex cases, we can still benefit from from OCT, and here is a case showing central atrophy here, as you see here by the hypertransmission of light through the cord. The limits of the atrophy are well shown here. You have what is called the ileum descent here, the external limiting membrane, one of the substructures of the photoreceptors. It ends here, and the other side as well. And you have something here that what was the basal laminar deposit persisting after disappearance of the retinal pigment epithelium, and you have the overlying disappearance of the photoreceptor layer here, but a persistence of the inner nuclear layer. So you can go very far in the histological characterization of the retina, even if you don't have histology per se. So the next step of the knowledge of dry AMD came with the onset of adaptive optics ophthalmoscopy. Here you have an illustration of the camera that we've been using here, the RTX1, and this is the SLO image of a case of age-related macular degeneration. The SLO magnification is here, and superimposed here you see the image obtained by adaptive optics ophthalmoscopy, and clearly there is a better contrast, better signal-to-noise ratio, and this enables in particular to distinguish the numerous melanin deposits that are present throughout the lesion, indicating that the RPE cells may not necessarily be dead, but they're dispersed and not organized the same way. And this is another illustration of a case of age-related macular degeneration. The optic nerve is here, and you have these lesions here and here. Another illustration here, the color fundus photograph here. This is not a color fundus photograph in this case. This is a infrared of the case shown here, and you see the improvement in the resolution offered by adaptive optics ophthalmoscopy. And this was coincident with the interest by histologists on the dispersion of melanin in dry AMD, which seems to be an important biomarker because melanin has the property of being a pigment that is poorly metabolized after its creation. I mean, it's a pigment that stays in the RPE and then can be phagocytized by macrophages, for instance, but is not metabolized by the retina. So it's a marker of the presence of RPE, and the pigment may stay in the successive cell and which it may transit. So it's a cellular tag that that you can use to follow up the progression of the disease, for instance. And different site of migration of these pigment spots have been described. Some related to RPE cells that are displaced, and some probably to inflammatory cells that phagocytize the RPE cells. This is an illustration of a case of a fovea, so it's a quite high magnification. The fovea is around, I think it's here, and you can see that there are several features that can be observed by adaptive optics ophthalmoscopy that are not visible elsewhere. You have this pigmented, presumed pigmented loaded cells over the way, but also something that initially was thought to be remnant cone mosaic, which resembles a lot to photoreceptor mosaic that can be seen by adaptive optics, but probably not the case. There is no histological studies that have shown that there are so many photoreceptor remaining after atrophy of the retina. So it's probably something related to sub RPE structures, or maybe basal laminar deposits, for instance. But that's our own hypothesis. We have no demonstration of this. On montage of adaptive optics, you can also navigate through the retina and explore the different features. And so it renders the figure of dry AMD even more complex than we thought initially. So we don't have to be afraid of complexity. And this is an illustration of the type of correspondence we try to make between adaptive optics imaging of melanin and the known distribution of melanin from histological work. And so for some of the melanin deposit that we observe, we can establish some hypotheses of what corresponds to what, but we have to say it's rather limited if we only use en face AO imaging. And I think correspondence will be easier done with if we use OCT as well. Okay, so, but there is another important and maybe the most important clinical feature of dry AMD. We did not really talk about it today. Is progression. I mean, it's having a snapshot of the image on one exam is one thing, but we have to understand what's happening during progression of the disease. And as I said, the disease progressed for years and years, very slowly, but slowly at that. And slowly in the fovea is for the patient maybe in fact very fast. So you may observe changes over years, and if you do time-lapse imaging of patients with dry AMD, this is an image taking I think over 4 years of the patient, and then you clearly see the dynamic underlying the progression of dry AMD. These are another case showing multifocal dry AMD spots, 1, 2, 3, 4, 5 spots. So see if spots of AMD may appear in different places. And this is accelerate, even each new spot, of course, accelerate the progression of the disease. And such imaging can also be done by OCT, as you see here. The central, the atrophy here is in the center, and you can see the progression of the lesion, of the atrophy, sorry, here, and the progressive increase of the hypertransmission through the cornea. So having a dynamic approach of the disease helps to understand probably helps to understand the cellular mechanism underlying dry AMD. This is illustrated here. It's a time lapse of adaptive optics, so at a higher magnification, and these pigmented spots, now you can see them actually moving. So there is indeed cellular migration also playing its part in the progression of dry AMD. And I think that was it. Okay, so this talk was to illustrate the fact that the fundamental knowledge of the disease of dry AMD progresses not only by the progress of histology and fundamental research, but also by clinical imaging. And so the combination of all these approaches is necessary to solve many of the issues related to this devastating disease. And hopefully from this, a new treatment will appear on the market. And it's hopefully this will contribute to achieve such objective. Thank you for your attention.
Thanks, Michel, for this talk. Yeah. If anyone has any questions, don't hesitate to put them in the question and answer just now.
How do I? What screen do you have? You still have my screen?
Yes, I think you need to stop your screen share so that we can pass over to Serge.
Got it.
Okay, do you hear me all right?
Your sound is quite quiet.
Is that better now?
Yep, that's okay.
Okay.
Okay, so Serge Meimon is going to talk about the physical basics of ocular imaging for non-physicists. Thanks, Serge.
Okay, thank you, Kate. Well, it's actually a tough one to talk about physical basics of ocular imaging for non-physicists because retinal imaging and ocular imaging is not that easy.
Your sound is very low.
Sorry?
Your sound is very low, at least for me.
Is that better now? Okay. So this is the outline of what I will present. Main concept in optical imaging, the challenges in the eye and how to address them. Some items about probing the phase and a summary that would be a zoology of instruments that hopefully will help you going through the rest of the conference. I see conversation. I hope that it's okay, that everybody hears me all right. Okay, optical imaging and main concepts. Some items about light. So light can be described either as a wave or as particles, which are small packets of energy we call photons. In terms of energy, we can quantify it either by the square amplitude of the wave, if you're in a wave approach, or by counting the photons. And about the phase, you can either talk about the phase difference between two waves, or talk about the photon travel time. That could be delta t. So these are equivalent notions, and you can talk either with one notion or the other, depending on what is the most suited to your purpose. Imaging. So you can either do some transmission imaging. You have a source, some medium and relay optics, an object that you want to image, then a medium and other relay optics, and a detector. Or you can do that in reflection, which will be the case in the eye. You have a source, some medium and relay optics, the object you want to image, and then you go back and detect what is reflected by the object. And the goal in imaging is actually to determine the transmittance or reflectance map of the object. This map can be, this reflectance, let's say, can be in intensity, the amount of intensity that is reflected, or in phase, meaning the phase difference that is induced by the sample or the object. Okay, let's, I will focus for some part of this presentation on intensity. So the optimal resolution, as was reminded by Gilles in his talk previously, is given, well, we talk about resolution, is lambda over 2NA in incoherent imaging. And I remind this formula, this lambda over 2NA. So NA can be seen also as in small apertures, it's F over D, with F the focal length of one lens, and D its diameter. The nice thing with this formula is that it goes back and forth. I mean, the smallest impact in the retina, or the smallest dot, you can focus on some journal paper with this magnifier. would be lambda f over d, or lambda over 2na. And the smallest detail that is detectable inside the retina would be also lambda f over d, or lambda over 2na. Okay, so that was one notion, and the other notion, it was the optimal resolution you can get. And there is another part that would be the optimal signal to noise ratio that you can get. And so now we'll talk about the photon noise or shot noise, which is some essential noise that you get when you do imaging, whatever the quality of your system. So let's do some practical experiment. How many cars are passing in the street every minute? If an average of 9 cars go every minute, so an average in your street, and if you do the experiment and you try to count them, actually you won't have always 9, because one will be a little ahead, another one will be a little behind. So let's say that you will have not 9, but 9 plus minus typically 3 cars that will pass every minute. If you have only 4 cars per minute in average, and you do the experiment, you will probably get something that is around 4, but you have 4 plus minus 2. And if you have 100 cars per minute, well, you will measure probably something like 100 plus minus 10. This fluctuation is the same you get with photons, actually. When you want to measure, when you have a flux that is 100 photons per minute, you will actually measure not 100 photon if you integrate with the detector for 1 minute, but you will get 100 plus minus 10. Actually, if your signal is the number of photons, you will have a fluctuation, which is a kind of noise, because it's an uncertainty, that will be the square root of s. If you look at here, 3 is the square root of 9, 2 is the square root of 4, 10 is square root of 100. So you have a signal s, a noise square root of s, and the signal to noise ratio is s over square root of s. So you get square root of s as a signal to noise ratio. This is the ultimate signal to noise ratio you can get if you don't have any other source of noise in your system. So the previous slide was about the optimal resolution. This one is about the optimal signal to noise ratio, or contrast, okay? So with a good sensor, if you have no additional noise coming from your sensor, 100 photons per pixel and per frame is fine, because with 100 photons, your signal to noise ratio is 100 over 10, which is 10. And a signal to noise ratio worth 10 is not too bad. It's actually fine. So do we get that in the eye? So I will talk now about the challenges in the eye and how to address them. And I will talk a little bit about photometry, aberration, motion, and scattering. So the first ophthalmoscope was actually designed in 1851 by Hermann von Helmholtz. It was the Augenspiegel. Sorry for the German-speaking people in the audience for my awful accent. And one drawing that was done with this instrument was this one, whereas a knife and this system of today gives that image. So it was not too bad in 1853. But actually, the goal now is not to have only these kind of images, but to reach cellular resolution. It was already, as was already exemplified in previous talks. So when you try to zoom in, you will try to reach the diffraction limit and to get the smallest impact, okay? And to get the smallest, detail detectable. So in the eye, the numerical aperture we have to consider with this formula is actually the numerical aperture of the eye itself, because it's the smallest numerical aperture in your system, in your optical system. It will be the limiting factor in your imaging. So I will take one slide that was, it was inspired by one famous slide by Austin Rorda, taking the diameter of the pupil of the eye. And you have the resolution that is given, with this formula that is very simple, with the size of the pupil, D, would be here. This is so the resolution you would get as a function of the pupil. So this is a curve, but it's easier to see it graphically. This is the size of the pupil. from 2 millimeter to 7 millimeter. This is the resolution you would get, the smallest detail you can grasp in your image. So with the 2 millimeter pupil, you would have a 7 micrometer resolution, and it's decreasing while the size of the pupil is increasing. So that's very nice, but actually, when you do the experiment, that's what you get. You should have something that is perfect and that is improving with the size of the pupil, but actually it's not improving. It's deteriorating because of the aberrations of the eye. And there are dynamics, as this animation is showing. So there is a way to tackle that problem. It's to correct for these aberrations in real time. And this is what is done by adaptive optics that was first implemented on the eye in 97, as early as 97. And this is what you get with the good adaptive optics. You get back the resolution, the almost optimal resolution that you had without aberrations here. Okay, this is for resolution. So now we're okay with resolution, but actually there is still another problem in the eye. You have the motion blur. You know, when you take the picture of a person that is moving very fast or too fast for your camera. So the eye is also moving really fast. This is a picture by Martinez Condé. Sorry, the reference is missing on that slide. But these are the drift, tremor, and microsaccades that you can witness on a retina. And the other moving points in your retina are, for example, the red blood cells that are flowing through your blood vessels. So if you want to have an unblurred image of your retina, you need to have a short exposure. And short exposure here means something that is shorter than 10 milliseconds, typically. Okay, so let's do it. We have an adaptive optics. We use short exposure and we take a picture of a retina. So 10 millisecond image with adaptive optics, and this is what you get. That is not as good as you might expect, because maybe there are some cells here that are hidden, but it's not very contrasted. So what happened? Why isn't that contrasted? So now we have to go into not so simple notions. We have to talk about photometry, scattering, and signal to noise ratio once again. Okay, so you remember I told you that with 100 photons per pixel per frame, you're fine. So do we have 100 photons per pixel per frame in our images of the retina? Actually, we're limited by safety standards, because when you're doing imaging in vivo, you don't want to send too much light in the eye. So we in France use the ISO norm, but the other standards used in the USA are giving pretty much the same amount of power. So if you're limited to 20 milliwatts per square centimeter at the cornea, which means that you cannot send more than 5 milliwatts in the eye. Okay, so 5 milliwatts over 2000 by 2000 pixels means that you have roughly 5 million photons per pixel and per millisecond. So that should be highly sufficient. But actually it is not, as I showed you previously. So what happened to these 5 million photons that entered the eye? So now I will talk a little bit about imaging in scattering media. For that one, I use some original slides by Gilles Tessier. So he used this image of Yosemite Park in California, which is nice to show the various kind of images you can have, or the various travel the photons can have in some scattering media. So let's talk about the foreground here. You have some detailed part of the image that you can see clearly and in focus. Then you have a more distant part of the image that is still clear but out of focus. And then you have some zone with the less and less clear zone where you're obscured by fog. It's a foggy part of the image. Okay, so now let's see what happens in the retina and what happens to photons actually. So these are all the little scatterers that you have in the retina and which you want to image actually. You are interested in only this part, which is the in-focus layer. This is actually what you're interested in. So what happens to the photons that are entering? There is a part that you're not that interested in, the photons that go through the retina and that are not scattered by all these scatterers, and that goes through its transmitted light. It's lost for you. Then there is light that is singly backscattered that will encounter just only one scatterer inside the in-focus layer. So that would be this path. This is your signal. This is what you're interested in actually. Then you will have light that is singly backscattered. That's nice still, but by out of focus layer. So that would be number 2. And then you will have the multiply scatter light. So these the one that have encountered a lot of scatterers, and that will create some diffuse haze. And I will so call this part the signal, here the intensity corresponding to single backscattering, but defocalized. And there is this diffuse haze that you obtain in the image. So you have all these components. So actually the image, the intensity that you get is the signal plus these 2 terms, 1, 2, and 3. Okay, and this is what you get in the image of the retina I presented before. So if you do some cut here and you plot the intensity, you will have some fluctuations that are actually your signal over some smooth component that corresponds to a defocalized component and the diffuse haze. Okay, so can we predict the shape and the magnitude of this diffuse haze? Actually, we can. So there will be a talk by Leah Kraft this afternoon by a model of the various layers in the retina that can help you predict what is the size of this haze and what is the ratio between these 2 components and the signal. Okay, but now when you have this smooth component here, why don't we remove it simply by data processing? Because it's not that difficult. You have some smooth component and you have some sharp components that are on top. With a simple data processing, you can remove these parts. So let's do that. And this is what we obtain. This is one typical image that you get. And what do you have? It's very noisy. So why is it very noisy? So this is imaging 101. Actually, I have to go back to this photo noise, shot noise slide previously. Let's say that among your 100 car per minute or your 100 photons per second, per pixel and per frame, you're actually interested in the 10 cars that are here, which correspond to the singly backscattered light. But actually you have all these white cars that are not interesting in your signal, but they yield their share of photon noise. And so then the signal is still S, but the noise is the square root of S plus the other terms that are here. And the sad thing is that the diffuse haze is actually much more energetic than the signal. And so the signal to noise ratio is actually something like signal over the square root of the diffuse part of the light. And so this is a lot lower than the square root of S that would be the ideal solution. So it would be nice if we could optically remove all these white cars, meaning remove all the photons that have been either diffused, multiply scattered, or the light that was singly backscattered but in out of focus layers. So this is what invented Marvin Minsky in 1953. The idea is the following: if you send light inside your sample through a small aperture, and you collect the light, you will still have all the components, meaning some singly backscattered light, some out of focus light, some diffuse haze. But at the way back, you add another little hole that will filter out these other components. You will just retain only what is at the same position in the same focus, so hence the name confocal, as the holes through which you have shined the retina. Okay, so that works well, but you do have only one point here. So you have to scan this point around all over your sample. So instead of having one frame as with a classical fundus camera, as I presented before, now you have to scan. Okay, so this was the original confocal scanning laser ophthalmoscope that was proposed in 1987. And here, these are drawings from this first seminal paper about that. And what is described here is actually that you're blocking with the pinhole the light that is multiply scattered or out of focus. And it was complemented with adaptive optics by Austin Arda and colleagues in 2002. Okay, so now we have removed these two parts thanks to the confocal pinhole. That's very nice. So there is still an issue in this kind of system. As I said, it's scanning. And when you have a scanning modality, you're not recording the photons of the whole scene at the same time. So if you have something that is moving really fast, faster than your raster scanning, you may end up with some artifacts. And actually the retina is moving pretty fast, or the red blood cells are rushing through your red blood, your vessels, your retinal vessels. So you still witness some part of artifacts, motion-induced artifacts in scanning systems. So to summarize, you have full field systems and confocal systems. Full field systems have a pretty, well, let's say reduced contrast, but they have no distortion and they're pretty fast. And confocal systems have a very nice signal-to-noise ratio and contrast, but they can display some distortion. In this a little dramatic example, this is actually the same zone of the retina. This vessel is imaged here, and you see that it's changing in aspect, not because it's actually changing in the retina, but because the person is moving really fast at that moment. So the question is, would there be an in-between between full field and confocal? Well, full field, you eliminate everything. Confocal, you eliminate just a small dot and you scan it. Well, we could think about a partial field ophthalmoscopy. So there is the line scanning ophthalmoscopes that are pretty interesting and starting to be used. And another maybe original idea is to use some other kind of partial field illumination, like with a checkerboard pattern. And this is what we have tried. And there is another talk this afternoon about that. So please check it out. And then another version that could be interesting, if you're able to project some checkerboard pattern in the retina, you could use some fringe pattern projection in the retina. And once you do that, you're able to do super resolution, because this projecting a pattern on a sample can allow you to do what is called structured illumination microscopy. In our case, that would be structured illumination ophthalmoscopy. And this is what has been achieved by Yan Lai team in our team. So with some nice super resolution images of the retina. So you can check out this archive paper. Okay, let's now go to another part of this talk. I only talked about how you can deal with energy, and I didn't talk about probing the phase. So, as I said, wave phase, which can be seen as a phase difference between two waves, or as photon travel times. So we said photon travel time. The light speed in vacuum is given by c. And in some medium, the speed is decreased. It's v equals c over n, n being a little being higher than 1, and n is called the optical index. In water, it's 1.33, and in most biological sample, it's around the same value. But n is linked to the density, and so when you have a lot of particles, you can imagine that light will travel a little slower. And so the density in a medium is linked also to the local temperature or the chemical activity that can change a little bit, slightly, the optical index of the cell, for example. And so when you are able to measure phase, which is linked to the travel time of the photon, well, I said that delta t is, the speed, is v equals c over n. So the travel time is the distance that is traveled divided by the speed. And so when you write that as, you write v as c over n, you get L times n over c. And this is proportional to L times n, which we call the optical path traveled by light. And it's actually a measurement of a travel time. And the nice thing is that when you access phase, you access the optical path. And so phase convey information on the position, because you're linked to the distance or the position, the path that is traveled by the photon. So the position of a scatter, the speed of the scatter, and also, as I said, the chemical activity, the local chemical activity in the retina, if you can sense the phase. So to sense the phase, you actually do coherent imaging, because detectors are not directly sensitive to phase, but you can manage by performing interference to be sensitive to phase difference between a wave and a reference wave. I won't go into detail about that, but just to say that you can use phase to bring actual resolution, and this is optical coherence tomography, or you can use it to bring information on blood velocity. This is what you do with Doppler holography. Or you can use it to bring cell activity sensitivity. And this is what is done on dynamic full field OCT. I try to give a few notions on the systems and techniques that will be presented throughout the rest of the conference. So to summarize what I've been explaining, let's do some zoology of instruments. So you have full field, partial field scanning systems, and you have incoherent system that are measuring only intensity, and coherent system that manage to extract the phase. Okay, so in full field, you have the flood illumination ophthalmoscope, also known as fundus cameras, also known as ANFAS imagers. Scanning systems, well, you have the confocal scanning laser ophthalmoscope. And the coherent version is the SLO OCT, so scanning laser ophthalmoscope optical coherence tomography. These three are the most used systems commercially or in research. Then let's add some coherent full field systems that are, there's a lot of developments on this system in our group, in the Paris group, the Paris Eye group, full field OCT, digital holography or holoscopy. For partial field, in coherent version, I would talk about line scan OCT, line digital Doppler holography, and a few systems on incoherent partial field system, checkerboard ophthalmoscope or line scanning ophthalmoscope. So this covers a lot, well, most of the systems you will hear about in the upcoming talks. So that's it, and I hope you will enjoy the rest of the conference, and that this tutorial will prove useful. Thank you.
Thanks very much, Serge. I think this was a really nice summary of the techniques that exist, and very much enjoyed the talk and the different animations. Does anyone have any questions here for Serge? Okay. If there's no questions there for Serge, the next session, I see a question's just come up, sorry.
Yeah, just one.
The question would be, what level of detail can now be imaged?
In lateral resolution, I would say it's around the micron. In vivo retina, that's the limit currently. I'm not sure, but I think that there's the beginning of using some super resolution techniques. 2-photon imaging could probably be used in the human eye, but there are still some problems of flux that is needed for that. So I would say that we're around between 1 and 2 microns. Don't see any other questions right now.
Not right now. Well, it was nice to have this introductory session to learn a little more about each other's disciplines before we carry on to the talks. So the next session will begin, we'll take a short break. It will begin in French time at 5:10, so in around 10 minutes. And the moderator for that session will be Ethan Rossi. And we'll be looking at imaging of photoreceptors and RPE. So see you in a few moments, everybody.